Ubiquitin got its name from ubiquitous, which means that it is everywhere. And I became absolutely obsessed with understanding how ubiquitin signals to control cell division.
What happened to us is that we scratched the surface and we found the same common molecular principle, which has been driven by ubiquity. Whether you want it or not, one day you will find yourself working on ubiquity.
Whether you like it or not, you'll find yourself studying ubiquitin. This saying comes from Aaron Ciechanover, winner of the Nobel Prize for Chemistry in 2004 for his discoveries regarding ubiquitin. Ivan Đikić even has it written on the wall in his lab. He and Brenda Schulman share a profound curiosity about ubiquitin, a protein which scientists have already known about for many years for its ability to mark for destruction those proteins that have become toxic or are no longer required. Brenda Schulman, an American biochemist, joined the Max Planck Institute in Martinsried near Munich in 2016 to continue her research on this protein, which occurs everywhere on the molecular level and is capable of deciding where, when and how certain proteins should be recycled. Brenda Schulman typically compares the role of ubiquitin with that of road traffic management, which calls for both precision and control.
Inside a cell, some processes need to be on, other processes need to be off, and ubiquitin is a signal that says off. My lab has developed ways, using chemistry, to basically freeze the molecular machines as if they are in the act of putting ubiquitin on to a specific target when it needs to go there. And so through this, we solve the three-dimensional structures that literally show how ubiquitin is placed in the right place at the right time. And these rules explain much of cellular traffic.
This process of target protein ubiquitination takes place in milliseconds. Several enzymes operate in a chain reaction. First, the E1 enzyme to start the process, followed by the E2, and finally the E3 enzyme, also known as ligase, which governs the transfer and binding of ubiquitin. It is this final protein complex, of which there are over 500 variants, that is of special interest to Brenda Schulman and her team.
So this structure here is one of the E3 ligase complexes that you've worked in the lab. And this is the E3 ligase that is acting as the final step to grasp the substrate and at the same time to basically put the ubiquitin onto the substrate.
E3 determines exactly where and when ubiquitin is placed. And if you think about this in terms of a landscape of traffic, you need to have ubiquitin, the stop signals, at the right place and the right time, and that is what is done by the E3.
Brenda has established many detailed mechanisms of how very large families of E3 ligases do their work. She's laid the foundation for understanding regulation and mechanism of a very large class of these E3s.
She has invented ways to get snapshots, basically, in these types of reactions. And without these technologies, we cannot solve important problems.
We're each experts in our fields. But then if it comes together, then, like I used to say, 1 + 1 is 3, and then you can really make things happen. So in the case of Brenda, when she studies the ubiquitination, we can quantify thousands and thousands of them at a time, and that gives her an advantage, of course.
And we hope to be able to actually see this inside cells, ubiquitinating this protein right here to see this regulation.
The next step for Brenda Schulman's team of researchers is to visualize the functioning of this E3 ligase in real time within our cells to better understand the close ties between ubiquitin and human pathologies.
So knowing the structural rules of how ubiquitin is naturally put on its target allows us then to understand how to manipulate this to direct ubiquitin onto disease-causing targets, and also which pathway we could regulate.
Further work into understanding the complex functioning of ubiquitin enabled Ivan Đikić to show that it was not just limited to marking proteins to be recycled and destroyed. Ivan Đikić is a doctor from Croatia who since 2003 has pursued his research at the Goethe University in Frankfurt, Germany. This is where his research teams focus on understanding the numerous roles and functions of ubiquitin.
Our first contribution to the field came out when we recognized that receptors which are on the plasma membrane, in order to be internalized and degraded in the lysosome, they need to be ubiquitinated. And ubiquitin is a driving signal that sorts them through the different compartments of the cell to be properly degraded. Then when we screened for many other proteins which can bind to ubiquitin, because we hypothesized that ubiquitin does so many functions because it's recognized by machineries which are involved in the different parts of the cells. So when we identified this large number of ubiquitin receptors, we actually read, like in a book, wow, the signature is from all different parts of the cells.
The signature, the stamp of ubiquitin present at different stages of cellular functions. A chain of ubiquitin regulating numerous physiological processes such as autophagy, which protects our cells.
Now we have found that ubiquitination plays actually a very important role both in autophagy, in Golgi regeneration, in mitochondria, and this is very good system that we are working on now, and this is in Legionella, from which we can indeed learn how it is hijacking multiple other signaling pathways.
Legionellosis, viral infections, cancer and neurodegenerative diseases are the targets for Ivan Đikić and his scientific team. All believe that ubiquitin has real potential as a treatment.
Ubiquitination is a perfect system in order to create new drug and modulate the different molecular signaling behind the cells. Imagine that you have a protein that is super essential for one cancer. If you block this protein, the cancer cells die. Then, using this proximity-induced drug, we can put together this protein with an E3 ligase enzyme, which causes the polyubiquitination and posterior degradation of this protein, and later will cause the death of the cancer cell.
In the last few years, I have been fascinated by the possibilities by utilizing modern technologies, including the proximity-inducing drugs where ubiquitin plays a role, mRNA technologies, nanotechnologies. I would like to use these technologies to now expand and apply the knowledge of ubiquitin to a little bit more therapeutical approaches.
These are clinical applications which Ivan Đikić follows here at the Buchmann Institute for Molecular Life Sciences in Frankfurt, an institution he co-founded and which is host to a new generation of scientists who combine basic research with biomedical development.
He's really one of the rare scientists on that excellence level to care for bachelor students up to postdocs and really create new ways of education for the new generation of scientists.
I feel that a lot of European science is team science, which means that by working together, the group can do more than an individual. Each of the students learns all of the other techniques, and each of the students has their project within the team, but they all influence each other's thinking.
When we train the new generation of students in biomedicine, they will be like physicists. They will be learning to share. As long as the mentors are preventing the openness, we are propagating the closeness of our labs. Science needs to be common, needs to be open, and then competition is not going to be a driving force of the students. It is going to be the progress.